Thank you, Takashi, and I'm sorry I started before I should have done. My apologies for that, but it is indeed a great pleasure and an honor to be speaking at the University of Hiroshima this afternoon. And I hope to see some cherry blossom, but maybe not in Hiroshima. Maybe I have to go to Tokyo for that this week. I'm going to talk, as you heard, about my life in science, and it's going to be in two parts. It's going to start with my early life, as you can see, when I got first exposed to science at school, then at university, and then when I began my research group. And then the second part will be an explanation of the work that was carried out in my laboratory, which did lead to the Nobel Prize that Takashi just mentioned in 2001. Well, I started my life as a baby, as most of us do. In fact, I think all of us do. And this is me. I'm not sure quite how old I am, but I think I look cheerful, don't you think? I wasn't born into a very academic family. I was born into a working class family. My father worked in a factory. My mother was a cleaner. And we lived in a very, very small flat in a place called Wembley. You may have heard of Wembley Stadium. Very good for football, Mr. President, which I know is of interest to you. And indeed, my brother worked in Wembley Stadium. We lived, and there were 7 of us, my parents, grandparents, brothers, sisters, in a small 2-bedroom flat. But unlike my rest of my family, who left school at 15, I was quite good at school and remained at school. And I became interested in science really at quite an early age when I was at primary school. And the first thing I remember today of my interest in science was when I read in the newspaper that Sputnik 2 would travel over London. Sputnik 2 had a dog. Inside it, a dog called Laika. It had been sent up by the Soviet Union. And just before it re-entered the atmosphere, the poor dog was killed, but just before it re-entered the atmosphere, it passed over London. And I asked my parents if I could stay up late, and I was standing in my pajamas in the front garden of my house, and across the sky came this moving star, Sputnik 2, very, very bright. I was extremely excited. I tried to chase Sputnik 2 down the road. I was running after Sputnik 2. It did beat me, by the way. And I was shouting to everybody who I could see and who could see me, look up there, there is Sputnik 2. And that was my first experience of what I could say is an interest in science. Then I got interested in natural history and astronomy. I had a little telescope. I looked at the changes in the seasons and the animals and the plants, and really thought that I would like to become a scientist. I did quite well in my exams and hoped to go to university. But I had a problem, and my problem was this, that to get into a British university, you needed to have a rather simple qualification, examination in a foreign language. And I have to tell you something else. I'm very, very bad at foreign languages. And I could never pass this examination. My school taught 3 languages, Latin, Ancient Greek, and French. I was too bad to even sit the examinations in Latin and Ancient Greek. So I sat the examination in French. I managed to fail this examination over a 3-year period 6 times, which I think is a world record for this qualification, and in fact could not get into any university as a consequence. And so I went and worked as a technician in a laboratory attached to the Guinness brewery. I'm sure you know Guinness, the beer. And I went there and I did the simple experimental work, making the media for the laboratory in which I worked. Actually, I did this rather quickly, so after only a few weeks, they let me play in the lab and do my own research project. And then the University of Birmingham in England, for which I'm continually grateful, looked at my application and decided to let me in without this qualification in a foreign language. I had to go and see the professor of genetics for 2 hours, and he said that he would get permission from the university administration, from the equivalent of the president. So the president let me in, but on one condition, and that condition was that I had to do 1 year of French at university. Luckily, I managed to pass the examination, which the French lecturer who taught me was the simplest in the world. I had to translate 500 words of scientific French into English, and the professor allowed me to take a dictionary into the examination. So even I could pass that examination. So I was interested in science. I did biological sciences rather than the physical sciences because I felt that biological sciences had lots of small problems, and that maybe as I was a small person, I would be able to solve the small problems, whereas physics, the problems seemed so big that I never felt I could be an Isaac Newton, but I might be a small biologist. And this was quite successful. This is me as a baby again. This is the most handsome picture I have in the entire series, by the way, so you should look at that one. This is what I looked like when I was an undergraduate, looking clearly at a flower, being a biologist. You will notice mainly that I had brown hair then, rather than white hair. Anyway, I decided I would do a PhD, and I wanted to work initially, when I went to university, on ecology, but by the time I'd got to the end of my course, I had too many unfortunate experiences collecting sea animals on the shore of coasts in a very cold England and Scotland that I wanted to work in a centrally heated laboratory. So I abandoned ecology and was interested in cell biology and biochemistry, and so did a PhD in biochemistry, examining how amino acids changed in fungi. I have to tell you, this wasn't a very interesting PhD project. In fact, I have to tell you, it's just about the most boring PhD project that you could imagine, because I thought that doing a PhD would be like doing all the best papers that I had read as an undergraduate. And really, I can tell you, it isn't always like that. So I grew my hair long, and I became a little wild. There you are. I had to put it in. That's really me, yes? And actually, this photograph was taken in a gliding club because I had other interests. And this is me gliding. You see the glider there just before I was going to take off. So this was what I was doing in my spare time as a student. And then I went and decided that if you were going to be a scientist, you had to do something that you were really interested in, really passionate about. So I thought a lot as a graduate student about what I should do once I had my PhD. And I decided that I should work on a very fundamental problem, even if it wasn't that interesting to the rest of the world. I really didn't care very much about what anybody else thought. And I thought the process of reproduction was basic for all life, and that you see the simplest version of that in the reproduction of a cell, because the cell is the basic unit of life. And when a cell divides from 1 to 2, and I shall talk more about this in a moment, then you are looking at the most basic example of biological reproduction. And I thought to understand how that process is controlled would be very important. So I went to Switzerland. I learnt genetics under Professor Urs Leopold. And then I went to Edinburgh under Professor Murdoch Mitchison and applied genetics to understanding the problems of the cell cycle. And in Edinburgh, this is a picture of me in Edinburgh, and this is one of my 2 daughters. This one is called Emily, and her older sister is called Sarah. Emily is now a professor of physics in University College London and works on high-energy physics at CERN with fundamental elementary particles. But once she looked just like that. My other daughter, Mr. President, is a television producer for football, and so gets to see all the World Cup games behind the camera. So I worked as a postdoc and then set up my own lab in the University of Sussex, studying the problem that I'm now going to tell you about. So that was the beginning of my life. Why did I say that? Tell you a little bit about it. It was to tell you how I became interested in science, to emphasize the importance of curiosity in how the world works, and passion in trying to work out what those problems are. And, most importantly, if you fail at certain things, do not get depressed or disappointed about them. Just keep fighting on, because you can make a success of it. And I failed quite often early in life, and it didn't really hinder me later. So that's the main lessons. So now I want to tell you about the reproduction of cells and how that is controlled. And that is the work that formed actually most of my working life, but particularly the first 10 years or so, which led to the work that led, as I said, to the Nobel Prize. So first of all, what is a cell? How was it discovered? Well, cells were first described by Robert Hooke. And Robert Hooke was one of the founders of the Royal Society in the 17th century. And he published a book in 1665 when the Royal Society was only 5 years old. And what he did is he took a piece of cork from a plant and cut a thin slice with a razor, put it under a microscope, such as the one that you see over here, and in it he saw lots of little boxes. And he called these boxes cells because they reminded him of cells in a monastery where monks live, all the little rooms arranged one next to the other, and so he called them cells. And here you see the picture, the very first picture of a cell. So he was the discoverer. He discovered it in Oxford. He didn't work for the university. He worked as the technician for a scientist. I live in Oxford at the weekends, and there's a plaque on the wall which describes the discovery of the cell by Robert Hooke. Now, the cell is the basic structural unit of all life, but it's also the basic functional unit of all life. And what do I mean by that? What I mean is it's the smallest entity of living things that exhibits the basic properties of life. Obviously not the complex properties that, for example, we as a human organism have, but the basic properties of being able to reproduce, to self-organize, to organize over space and time, to be homeostatic. All these processes are found in the cell. And this idea had its origins in another scientist, this time a German scientist in the 19th century, Rudolf Birkhoff, who founded the medical science of pathology. He was a medical doctor. And he said in German, this sentence here, "Every animal is a sum of vital units, each of which bears in itself the complete characteristics of life." That's a complicated sentence. It simply means that the cell is vital, it's alive, and it has the characteristics of life, the simplest unit of life. He also said in Latin, but translated here into English, that each cell arises from a pre-existing cell by division of that cell from 1 to 2. So if you put these ideas together of Hooke and of Birkhoff, you have the basic idea of what a cell is. And I'll show you a dividing cell. This is a human cell. It's a mammalian fibroblast, that is a cell type that is found in humans, which are studied very often in the laboratory. And you can see this cell dividing in two, having just reproduced itself. And this is the process, the control of this process, which I wanted to understand. Now, why is understanding of this process important? Well, there's 3 reasons I want to explain briefly to you. The first is, as I've already indicated, that it is the basis of growth and reproduction of every living thing. That if you want to understand how we grow, if you want to understand how we reproduce, and how every other living thing that you can see, it doesn't apply in quite the same way to bacteria, for example, but every other living thing, then you need to understand the reproduction of a cell. So it's a very basic, important biological problem. The second one, which is a little bit more difficult to appreciate, but it really is the next chapter on from Watson and Crick and their idea that DNA copies itself, and that is the basis of heredity, and that is, of course, called DNA replication. Now, what Watson and Crick did was to show by establishing the structure of DNA with Rosalind Franklin and Maurice Wilkins, working in Cambridge and London, what they showed was that the structure of DNA is such that you can understand how DNA is copied. But that is a chemical mechanism. It only makes biological sense when it is found in cells and exhibits its properties in cells. And what you need to understand is how that process of copying DNA occurs in a cell and how each of those DNA copies ends up in the 2 daughter cells. And it's the control of those processes which I wanted to understand, how you can link copying of DNA into cells. And of course, the third reason why it's important is that cell reproduction is critical for cancer for 2 reasons. One is that cancer is caused when cells divide out of control, reproduce out of control. And the second is, because as I shall briefly explain in a moment, that it is a consequence of genetic damage to genes, which causes those cells to divide out of control. And that damage to genes is very closely linked to the process of DNA copying and segregation that occurs every time a cell reproduces. So just briefly, to go over those 3 points, it's the basis of all growth and reproduction. You have a single cell, it divides by repeated divisions. This is a mouse embryo, we look not so different, and each of these dots is a single cell. So it's the basis of the formation of that embryo. This is a very important picture. It shows sperm trying to get into a single cell, and I show it in case anybody in the audience is getting bored with my talk, because I want to remind you that every one of you in this audience was once a single cell. And if you're not interested in that, then maybe it's the time now to leave. But every one of us was a single cell, so we have a reason to be interested in cells. The second question, which was how DNA copying is linked to the cell and the cell reproduction, is shown in this simple figure here, where this is the cycle of cell reproduction. It's called the cell cycle. DNA is copied during what we call S phase, the synthesis phase. Prior to that is a period called gap 1 or G1. After it is a second gap, gap 2 or G2. Then, and you see the replication is these two blue bars, is at M phase or mitosis, the copied DNA segregates into the two newly divided cells. So it's the linkage of the chemical copying of DNA into eventually the copying and segregation of those chromosomes into two newly divided cells that is key to this process and important for cell reproduction. Third one, importance for cancer. This is it could be a thin layer of cells in our body which are dividing and growing normally. Then a gene becomes damaged, that's the red cell here, so it now divides out of control, so it's no longer a very orderly structure. It now forms many cells growing out of control, and this is the basis of a tumor. So this is the basic property of all cancer. So those are the reasons why we should be interested in it, and this is the problem I wanted to investigate. How is it controlled? How can we investigate it? Well, how can it be investigated? And this is the problem. I was 24 years of age. I had my PhD that I wanted to take on. And I learned something about the use of genetics to look at the cell cycle, which had been done by Lee Hartwell, who was mentioned a little earlier and got the Nobel Prize too. And he used, and I used, a genetic approach to study this problem. And I've drawn a figure here to illustrate the approach. Because nobody knew at all how the cell cycle, how cell reproduction was controlled. We had no idea. You couldn't even guess how it might be controlled. But you can get at it by the use of genetics. This is a yeast cell. I'll show you a real picture in a moment, because I'm going to work on the fission yeast, because it's very simple. And within this yeast cell, there are 5,000 genes. Here you see about 10 of these, each one of these green blobs. What you can do here, and what I did, was to treat these cells with a chemical that would damage DNA. We call that a mutagen. It would mutate or damage the DNA. And you would randomly damage any one of the 5,000 genes in the yeast cell. Now, if the yeast cell can complete the cell cycle, it will reproduce. You see these lines here that will divide the cell. If, however, you hit a gene and damage a gene that is necessary for cell reproduction, the cells can continue to grow, but they can never divide. And I'm going to show you pictures of that. So that was the first experiment I did, and I identified 30 CDC genes, that stands for cell division cycle genes, 30 genes. And that was done, and that's the paper that actually Takashi Toda described, I published in 1976. Since then, we have sequenced fission yeast. We have deleted, in my laboratory, every single 5,000 genes, and we now know there are around 300 such genes. So at this time, we had only isolated 10% of the total. But it was a start. It's a start to the problem. It defined genes that were needed for cell reproduction. This is what the yeast looked like. This is a picture in an electron micrograph of fission yeast which is different to budding yeast. Budding yeast is much more useful. It's used to make sake, it's used to make bread, it's used to make beer. All fission yeast does is contaminate those cultures and makes it worse. But it's good for science. This is actually the first picture I can find of a fission yeast. It was contamination in Carlsberg's lager in Copenhagen in 1880s. Now, I've described how we can isolate mutants that are defective in genes required for cell cycle. And I'm just going to illustrate it here. This is a normal cell with a nucleus in it, with the DNA in it. It grows, increases in cell size, it divides into two, it goes back again, and it keeps doing this. If, on the other hand, you are altering one of those CDC genes, they continue to grow and produce this cell cycle blocked cell because the CDC gene is defective. This is the paper that Takashi mentioned, which I published with two collaborators, Kim Naismith, now Professor of Biochemistry in Oxford, and Pierre Thuriot. And this is what wild-type yeast cells look like, fission yeast cells look like, and you see the elongated ones that can't divide. It's very simple. You can see it all with your own eye under the microscope. But there was a problem here. What this did was to identify genes that were necessary for reproduction. It didn't necessarily identify genes that controlled reproduction. And for that, a different sort of mutant was required. You've already seen this figure. This is the normal cell cycle, let me remind you. Now, imagine what is a control. A control is something, there are different definitions, and I'm going to use a second definition a little later in my talk, but one idea of control is something which is rate limiting for progress through the event that you're interested in. In other words, determines the rate at which the cells can reproduce themselves. And I say it's a little bit like if you have a car and you have the accelerator that makes it go faster, that is rate limiting for the speed of the car. And it's the same logic here. Imagine now a cell which normally does this, but actually you have a mutant in a gene that makes it go faster through the reproductive process. Then what will happen? It's the complete opposite to this. These cells will divide before they have reached the normal cell size. They will be advanced prematurely into cell division. And that will produce cells of a small size. And because I isolated these in Scotland, in Edinburgh, and because wee is the Scottish word for small, I call them wee mutants. I thought it was very funny at the time. 40 years later, it's no longer quite so funny. So be careful when you call genes funny names. Just imagine you're still talking about it 40 years later. So these mutants identify rate-limiting steps. And here is an example of the first wee mutant published in 1975 when I was in Edinburgh. And I was helped in thinking about it, my friend Peter Fantes. These are wild-type cells. These are the wee mutant cells. Now, the way I told that story isn't quite accurate. I told it as if I'd sat down, thought about it, and looked for wee mutants. It isn't true, I'm afraid, ladies and gentlemen. I just was looking under the microscope, and I saw cells that looked like this, and I wondered what could be causing it, and then I had a little light bulb flash in my brain, which said these cells are advanced prematurely through the cell cycle, therefore they define a rate-limiting step. I mention this because it's important, particularly in biology, to follow where nature may lead you, because you may not always think of all the things that can happen, but following nature shows already that something is happening that is interesting. So I proceeded to look for these mutants, which were difficult to isolate. I spent over 1 year collecting 50 of them to see how many rate-limiting genes there might be. I got to mutant 48, and they all were mutations in WEE1. So I was getting a little tired of this. And the 48th mutant, I isolated on a late raining afternoon on a Friday, and it was covered with a fungus on the plate. So I threw it away in the rubbish. I went home, I had my tea, and then I felt guilty. And so I came back on my bicycle, back to the lab, and fished it out of the rubbish. And I then spent the next week or two cleaning it up. And that mutation was the only one that wasn't in WEE1. It was in a gene I called WEE2. So now I had two genes that controlled the rate of progression through the cycle. They both controlled when mitosis took place. One of them, WEE1, was an inhibitor. It inhibited the process of going through the cycle. WEE2, because it was very rare and because it was dominant, which I won't explain for those who are not familiar with it, I speculated was an activator that accelerated cells through the cycle. I did some fancy genetics I'm not going to explain to show that WEE1 was an inhibitor, had something to do with it being a nonsense suppressor. This is what I looked like at this time, and this is my friend Pierre Thuriot. You can see it's clearly the 70s. Then, we had two genes that were controlling entry into mitosis. I was excited by this, but the world wasn't terribly excited by it, and I found that I couldn't get a job. I applied for jobs as a professor, assistant professor, lecturer, and I failed to get them. Because everybody thought that the cell cycle wasn't controlled here, which is what I was arguing, but was controlled here at the beginning of the cycle. So Lee Hartwell, who I've already mentioned, had defined a control here by using CDC mutants that blocked at different stages through this reproductive process, and then asking, could those mutants that blocked in different places, could they undergo another developmental pathway? And yeast cells don't do many developmental pathways, but one thing they do is conjugate and then undergo meiosis and sporulate. So I copied what Lee did and just blocked with my 30 CDC mutants at different stages, challenged them to undergo conjugation, because if they became committed to the mitotic cycle, they wouldn't be able to conjugate, but if they were blocked before it, they would be able to conjugate. A simple experiment. I did the whole series, and I found one gene that blocked here, called CDC10, where cells still could conjugate, and all the rest could not. And I left till last the control experiment. And the control experiment was to look at the CDC2 mutant, which was my most studied mutant, because CDC2 turned out to be the same gene as WEE2. So I'd shown that CDC2 controlled entry into mitosis. Now, if you can still conjugate, you'd expect 80 to 100% of the cells to conjugate. If you couldn't conjugate, you'd expect 0 to 5% to conjugate, just background. And when I did the experiment with CDC2, I constantly got the same answer of 25%. And that's neither one thing nor the other. And this was depressing because this undermined the whole experiment. You know, how could you get something that was 25%? So I kept repeating it. It needed very precise temperature control. So I bought a bigger thermometer to make sure the water bath was the right temperature. I still got 25%. I put the work away. I left it for a few weeks. I did it again. I still got 25%, and I didn't know what to do. And then I woke up in a dream. And in my dream, I had on one shoulder an angel and on the other shoulder the devil. And the devil was speaking to me in this ear, and it was saying, you know, Paul, this piece of work is complete as a paper, a research paper. The only result that doesn't fit is CDC2. And do you know something else? You're the only person in the world who knows that you've done that experiment. So I, as the devil, suggest you throw it away and publish everything else. And then you can apply for jobs. You will fit in with what everybody thinks, that it's controlled here, and you'll get a job and everything will be fine. Then the angel whispered into this ear and said, Paul, you cannot do that because science is the pursuit of truth. And I know that if you try to publish this, the reviewers will reject it. And that means you won't be able to publish your paper. And that means you won't get a job. And that means you and your 2 babies will starve and have nowhere to live. But science is the pursuit of truth. So I woke up in a cold sweat. Would you not wake up with a cold sweat, faced with that choice? And then I, like a politician, thought there had to be a third way. A third way. And the third way was, what if 25% was the right answer? And why I tell you this story, which I'd hoped somebody would laugh at, actually, because I was trying to make it funny, why I told you that was that all this time I thought I'd got the wrong answer. And I never thought what it could mean if it was the right answer. And so I thought about it for a few days and realized what if CDC2 not only was required here, but was also required here. This is quite a short period, only maybe 15% of the cycle, and if all these cells conjugated, it could give you that answer. And that was actually the case. So now, CDC2 was rate limiting for overall progression and controlled onset of mitosis, and was also responsible for commitment to S phase. So it was necessary at the 2 major controls in the cell cycle. So I published this paper. I still didn't get a job, by the way, not for a while. But I now realize that it was key to find out what CDC2 did. So how did we do that? It was around 1980, and gene cloning was just beginning. So I went on a course and learnt how to clone genes. And this allowed me to do this experiment here, which I drew at the time. All these are original figures. This is a fission yeast cell. This is defective in CDC2. The cells cannot divide. I put into it a library of fission yeast genes. If one of them contained an intact copy of this gene, then it could produce that protein. It would rescue the defect, and the cells could divide and form a colony on the plate. I could then isolate back this DNA, which was on a circle called a plasmid, and identify the gene, which is CDC2. So I devised these techniques that allowed this to be done, isolated the gene, which was, for those who in the audience are biologists, was 2 kilobases in length, 2,000 nucleotides. It then had to be sequenced. And I went to Fred Sanger, who got 2 Nobel Prizes for DNA sequencing. He put me in contact with his postdoc, Dr. Hindley, and he sequenced the gene, and it took him 15 months to sequence 2,000 nucleotides. We can now do it in maybe 15 milliseconds on the machines, but it took him 15 months. And that showed that this gene, when we sequenced it, it had only 1 hit in all the genes that had been sequenced, because there were only about 100 genes that had been sequenced at this time, and that was an oncogene called pp-sarc60, the Sarc oncogene, and it was thought by the virologists who worked on it that that gene encoded a protein kinase. Protein kinases put phosphates onto other proteins, changing their properties. So I guessed that it was a protein kinase. I expressed it in bacteria, this gene. I made protein. I put it into a rabbit. I made antibodies. I used the antibodies to purify these proteins and then did protein kinase assays. And it was a protein kinase, and this is how it works. A protein kinase here, this is a phosphate, which is a negatively charged molecule. It's put onto the protein, which changes the properties of the protein. In this case, it allows substrates to get in. And the idea is the protein kinase phosphorylates the many substrates that are required for S phase and M phase. And that's how it works. This actually was the assay done by Sergio Moreno and Viestas Simonis measuring protein kinase activity. And this is what I looked like then. Okay? I seem to look strange for most of my life. I don't quite know why. So we cloned the CDC2 gene. This was in cloned the gene, showed it was a protein kinase, showed it changed through the cell cycle in papers in the 1980s. And we also cloned a number of other genes that were required that interacted with CDC2. There was CDC13, which encoded a cyclin, and cyclin was discovered by my friend Tim Hunt, who also got the Nobel Prize, and we showed that this was similar to cyclin from sea urchins. We cloned WEE1 and showed it was a protein kinase that inhibited CDC2. We cloned CDC25, which acted as an activator of CDC2, and we proposed that was a phosphatase, and that this whole network here was controlling the entry into mitosis. Now, I was pleased with this work, I have to say, but once again had a problem with whether the rest of the world thought this was interesting. And frankly, this was yeast. And most people are not really interested in yeast. I just have to admit it. I'm interested in yeast, but most people aren't. So I realized that we had to show, if it was possible, whether there was a CDC2 gene in more interesting organisms, particularly ourselves. So I tried to persuade people in my lab to look for the human CDC2 gene. This is long before humans were sequenced, so we couldn't look for sequence. Well, we tried conventional methods, and they didn't work. The person working on it, I'll show you a picture of her in a moment, was Melanie Lee. And then we had a wild idea that we could use the same technique that I've already shown you. You remember this, and a gene library that we made from fission yeast. And I got a gene library made from human cDNA, so basically human genes, that had been constructed by Hirota Okiyama, who worked for many years in Tokyo, when he was working in Paul Berg's lab. And they generously gave me this library, and so I put the human library, together with Melanie Lee, into a CDC2 mutant. Now, what were we looking for? We were looking to see whether there was a human gene that could do the same job as the fission yeast gene that was defective. Because if it did, then the cells would grow and divide, and we could isolate the plasmid back, and it would have the human gene on it. This was a bold experiment, because the last time that humans and yeast cells shared a common ancestor was around 1,500 million years ago. That's a very, very long time. And what we were demanding was that the control would be still so good and so similar that taking the gene from humans and putting it into yeast, it would work. And furthermore, we were fishing amongst the 25,000 human genes to find one that might work. This experiment had no right to work, but it did work, which allowed us to isolate the human gene. I was enormously excited. This is Melanie Lee when she did that experiment. We did it in 1987. And when it was sequenced, and it was very, very tense time when we were sequencing this gene, we ended up with 61% identity between the human gene and the fission yeast gene. So despite 1,500 million years of divergence, these genes are very, very similar, and you could substitute one with the other. And we therefore concluded that the way that the cell cycle is regulated is in the same in yeast and in humans, and therefore is the same in probably every eukaryotic cell, that's animals and plants, that we can see. I want to show you a picture which I took of the first clone of fission yeast cells defective in CDC2 being kept alive by the human CDC2 gene. So how do we think all of this works? It's very simple. More recent experiments have allowed us to simplify these views, and I'm just going to explain the conclusions without explaining how we've shown it. And what happens is, as cells go around the cycle, the level of CDC2 cyclin-dependent kinase increases. And I've shown that by increasing amount of redness. To undergo S phase, you only need a low level of activity because the substrates required for copying DNA can be phosphorylated by very low levels of the enzyme. Strangely, when you get more activity, you inhibit S phase, and that means you only have one S phase in each cell cycle, important for genomic stability. When the activity rises even higher, then you get enough activity to phosphorylate all the substrates that are required for mitosis, and there's about 10 times more required here than are required here. When cells exit mitosis, they degrade cyclin, that's what Tim Hunt showed, therefore you kill all the activity and you start again. And this is the simple model that can explain the cell cycle. In many, if not all organisms, the distribution of these different thresholds is provided by different CDC2-like genes, what we call different cyclin-dependent kinases. But we've synthesized a strain where we can do it all with a single gene, which is why we can simplify the model. So, to conclude, cyclin-dependent kinase, of which CDC2 is an example, controls the cell cycle. Its increasing activity drives S phase, blocks S phase, drives mitosis. And that is the key way in which cell cycle control is imposed. Secondly, this basic control, we argue, is the same in every living thing, and it's been conserved for over 1,000 million years. So essentially, it arose, I think, with the origin of eukaryotic life. It was so central to eukaryotic life that it remained unchanged since then in all living organisms. Well, I don't think this level of conservation was expected by the community. Certainly when I was working on yeast, those working on humans thought that yeast was a waste of time. I suppose even I thought it might be a waste of time, but it wasn't the case. However, I went back to the literature and I found a quotation from Theodor Schwann. Schwann was one of the two German scientists that in 1839 proposed the cell theory based on Hooke's work at the time and what was happened since then. I'm going to read it to you and then interpret it. We have seen that all organisms are composed of essentially like parts, namely of cells. That was Hooke's point. Everything is made of cells. Secondly, what Schwann said, these cells are formed and grow in accordance with the same laws. In other words, he postulated that cells grow and form subject to the same processes. He thought they would be conserved. Hence, that these processes must everywhere result from the operation of the same forces. In other words, he was struck by the bold idea that the control of reproduction, the control of growth, the control of reform is likely to be very conserved in all living organisms. It just took us another 150 years to show that for cell reproduction, he was completely right. Thank you very much, ladies and gentlemen, for listening to me.
皆様お待たせいたしました。
Thank you very much
そ
for
れで
your
は
waiting.
ただい
Now
ま
we
よ
would
り
like to start
パ
the
ネル
panel
ディスカッション
discussion.
を行います。本日ディスカッションを行っ
The
ていただ
panel
くパネリ
discussants
ストの先生
are
方
to
にご
be
入場いただきます。どうぞ拍手で
get
お迎えく
into
ださい。
the stage. So please welcome
パ
them
ネリ
with
ス
your
ト
applause.
入場。
All the panelists are to be on the stage.
それではパネリストの方
Now
の
we
ご
would
紹
like to
介をさせ
introduce
て
the
いただきます。
panelists.
パネリスト
The
お一
first
人目は
panelist
先ほどご
is
講演いただき
the
ました
speaker
イギリスフランシス・クリック研究
who
所
is
所長
the
ポー
director
ル・ナ
of
ース先
the
生です。
Francis Crick Institute, Sir Paul Nurse.
続いて
Next to
ナー
him,
ス先生のお隣にいらっしゃいますのが独立行政法人
the
種類
president
総合研
of
究
the
所
National
理事長
Research Institute
後
of
藤奈美
Brewing,
先生です。
Dr. Nami Goto.
この度後藤先生は
Dr.
自ら
Goto
活力向上を先導した女性として本
has
学のメタセ
been
コイア賞を受賞されました。
awarded the Meta
後ほ
Sequoia,
ど表
who
彰
is
式を行わせていただきます
due to her great work. So we are going to have
続き
the
まして
awarding
医師
ceremony
薬保健
of
学
the
研
Meta
究家
Sequoia.
研究課長
And
教授
the next to her,
安井渡先生
the
です。
Dr. Wataru Yasui, who is the Dean of the Graduate School of the Biomedical
医
Health
師
and
薬保健
Science,
学研究科教授
a professor
Chayama
of the
Kazuaki
Graduate
先生です。
School of the Biomedical Health and Science, Dr. Kazuaki Chayama.
続いて向かって
Next,
左手側の
on
机
your
にいらっしゃる
left
先生
side,
方をご紹介いたします。一番奥にいらっしゃいますのが理学研究科研究課長教
the
授
Dean
立井真一先
and
生です。
Professor of the Shinichi Tate at the Graduate School of Science.
そしてパネリスト最後
And
の
then,
お一人は
and
生物圏
lastly
科
but
学研
not
究
least,
科副研究課長教授三本木義弘先生です。
Dr. Sambongi Yoshihiro, the Vice Dean of the Graduate School of Biosphere
なお、ファシリ
Science.
テーターは
And the facilitator of this panel discussion is the specially appointed Professor Toda at the Graduate School of the Advanced Science of Medicine, or
そ
of
れ
the
では
matter,
ここからの
rather.
進行は
Now
戸田
I'd like
先
to
生
hand
に
the
お願いした
microphone
いと思い
to
ます。
Professor
よろしくお
Toda.
願いします。
Professor Toda, please. Thank you very much.
それでは今から40
Now,
ぐらいですか。
we are going to have about a 15-minute talk, and together with Sir Paul Nurse for a panel discussion. We prepare several topics for discussion, and we would like to invite the Sir Paul Nurse opinions for this agenda. First, actually, Professor Sambogi at Oxford, that was in 1992, or for several years. He has been conducting research at the same time together with the Sir Paul Nurse. So
I prepared English talk today, but we decided we should talk in Japanese. Is that okay? I'll use Japanese. I'm sorry. これ立った方がいいですかね。いや、見えなかった。私、先ほど戸田先生からご紹介ありましたように、
as I was introduced by Dr. Toda, I have personal experiences with Sir Paul Nurse. Just several minutes, I would like to introduce what kind of experiences that I had. And this explains about the Department of Biochemistry of Oxford, and this is the pamphlet of that. And this is the actual one. And the reason why I'm keeping this is as follows. In starting from 92 to 95, I was at the University of Oxford at the Department of Biochemistry, and I was at the postdoc, and I studied there for 3 years. Unfortunately, I'm not at the same lab with him, but Sir Paul Nurse is quite famous, and I've seen him quite often at Oxford. And this is the page 21 of that pamphlet. The young Sir Paul Nurse, a picture is shown and his research is introduced there. He mentioned that he used to have the brown hair, and actually at that time when I met him, his hair was brown. And in this way, I had the chance to meet him. And that was about 25 years ago, since I was at the Oxford. That's why I've got this pamphlet. I know him very well, and I'm not sure that he remembers me or not, but I had a lunch together, and he mentioned that he remembers me, so I was surprised that he still remembered me. And in 92 and 93, this is a summary of his work at that time. And this figure was already shown in his presentation, starting with G1, S, G2, and M. And there are the two places that the CD2 works. That was explained by him. And actually, the figure has been already written down. And this portion, this is the memo that I've got at the lecture, G1, S, G2, and M. So in a clockwise manner, the same figure is shown in my notebook. And it is a blown up of that in 1993 at May 24th. So that is 24 years ago, by participating in the seminar organized by Dr. Paul Nurse. And the message that I want to deliver today, this is message 1. I've got a very important message from Sir Paul Nurse. At the very beginning of the seminar, Sir Paul Nurse says that I'm going to talk about unpublished talk. So I was so excited by knowing that he is going to talk about unpublished work. He's very famous, and I had a chance to know his work, which is not published yet. So I was very excited. And then I was listening to him during the seminar. And I was at the postdoc, so I obtained the PhD and went to UK, and I had to organize a seminar by myself. So that was at my position. So at the very beginning, at the Oxford days, I've got his comment that he is going to mention unpublished work, and he tries to draw the attention. But it's quite effective to me. I was very triggered and interested in that. So at the end of the seminar, another important message is given from him to me, and the key element, molecular biology, biochemistry. Let me explain. The study of the biology, what is the important points of the biology is the molecular biology and biochemistry. So these two different approaches should be combined for conducting the study of the biology. That's what he taught me. Molecular biology, well, the high school students may know this. This is this biology, but biochemistry is chemistry. So biology and chemistry, there are two different classes, but at the university, they are getting closer. And with these two approaches, the new science would be created. So by listening to his talk at that time, by combining the different approaches to seek for the one science, I learned from him, and I selected to study in the field of the biochemistry. And he talked genetics a lot today. Genetics is the biology. So this is the approach to the biology with the gene. But the cloning of the gene and give the expression and to activate the kinase, and that is the side of the biochemistry. So these two approaches are also combined together within his project and his study as well.
受け手としてミノルにある
So,
科学教育のためのヒントをここで簡単にまとめて
we would
私
like
の
to
紹介とさせてい
summarize
ただきます
some of the
まず一つ目
hints
は
or the points
いい科学教育を
to
受けるためには今日のよう
for
ないいセ
the
ミナーをに
fruitful
参加するとい
science
うことがまず大事だと思います。
education
そういった意味で
for
ポ
the
ールナンス先生には
recipient
今日本当に感謝
that
して
we
おり
have
ま
to
す。
join to the good seminar like
で
today.
それから
I'm very grateful to have this
2番目
seminar.
としましては、話
As a
し手
second
のメッ
point,
セージを
that you have
捉える
to
というところですね。
catch the
私
speaker's
24年前
messages.
先生の話を
24
聞い
years
て
ago,
2つ
by
のメッ
listening
セ
to
ージを得て非常に感銘を受けました
I've
そ
got
して
two
今日たっ
messages
たまあ
and
15
it
分
was
ぐらい
so
前ですか
impressive.
もう
Just
24年
15
ぶりに
minutes
先生
ago,
の話を聞い
in
てまた
24
新しい
years
メッ
absence,
セージを受
I've
けてまた
got,
感銘を受けました
again, I listened
ま
to
あ
him
人それ
and
ぞ
I
れ皆さんそ
was
れ
so
ぞれ
impressed
感
about
銘を
his
受けた点
talk.
があるかと思いますが
The,
私一つ挙げれば最後の
One
シュワ
important
ンが
point that the
1839
Schwann
年
in
に先生が
1939,
証明されたことを
the
先見の明
thing
ですでにもう論文に
which
書い
has
ているとい
the
っ
Sir
たことです
Paul
ね。
already
私にとってシュワンっていうのは
reported
発酵
has
学の
been
分野の先生なんですけれども
mentioned by
あそうかシュワン
Schwann
ってそういうようなメ
before
ッセージを
that.
残してたって
So
いうことだ
he
けを
is the
勉強できる
expert in
ですね。
the fermentation,
それから最後
but
三つ目
he
この
has
Go
the
analog
deep
アナログ
insight.
ですが
And the third point is the Go analog.
こう今すごい
Well, these
IT です
days,
か
the
が流行ってい
IT
ますけれども
is
手書
prevailing
き
the
で
world,
メモを取るというのは
but
大事だなと思い
handwriting
ます
and handwritten notes are very important.
今日ポールナンス先生の講義でも何
By
枚か
listening
手
to
書きの
his
スライドが出てきたかと思いま
talk
す。
and
で
in
それ
his
見るとなんか重要
presentation,
な
you
スライドは
mentioned
みんな手
his
書きだったなっていう
handwritten
のが
notes.
分かります。
Important points
それ
are
で
always
若い学生さんと
handwritten.
か、まあ、もちろん高校
So
生
young
もそうなんですけれ
students,
ども、
including
スマ
high
ートフォン
school
でこれをカ
students,
シャって取るのはやめましょう。
that don't
手書
take
きで書いた方
those
がいいと思います。
slides
そうでなけれ
with the
ば
smartphone
これ
or the
写
camera.
真で撮ると忘れてしま
You
い
have
ま
to
す。
take the notes.
です
If
の
you
で
take the
手
picture
書きで
by the smartphone, and
いう
then
ことが大事
easy to
と思います
forget.
これ
But
が
once you
私の
write by yourself,
ポー
then
ル
you
ナンス
can
先生と
remember.
の思い出です。
So this is my memory with Sir Paul
ありがとうござ
Nurse.
いました
Thank you very much, Dr. Toda, to giving me this opportunity.
すいません。
Well,
もう一点
sorry,
もう一点
one more point. One
も
more
う
point,
一つ
sorry.
ここに Go analog without smartphone と
Go
か言って
analog
ますけれど
without
も、
smartphone.
このセミナー終わった後、ポールナン
Well,
ス先生の写真撮るのはいいんじゃないかなと思います。
This seminar, I think it's okay for us to take pictures together with Sir Paul Nurse, just as the memory. It is okay to use the smartphone to take pictures with him.
A couple of points. すみません。
It's a multidisciplinary research.
Please interrupt panelists, please. Maybe you can tell us or talk about, Francis Crick perhaps.
Yes, so I apologize for speaking English, but you should be grateful I'm not trying to speak Japanese. That's all I can say. The multidisciplinary research is something that is talked about by a lot of people, but it's actually very difficult, I think, to do well. And I think we should all start with exactly that statement. It is difficult to do well, and you have to work hard at doing it. The point you were making here is a simple example of the need to approach biological problems in different ways and using biochemistry, molecular biology, and genetics, because they can correct each other because they approach things in different sort of ways. It's a simple example of it. But it is important because often geneticists don't do very much biochemistry and biochemistry don't do much genetics, and it's good to put those together. If that is quite difficult to do well, it's even more difficult to get those who are trained in different disciplines to work well together. I'm thinking of biologists working with chemists and with physicists or mathematicians, computer scientists. And I think something that is really, really underestimated is what I call the sociological problems with that. That is to say, if you are trained in a particular way, as a biologist or a physicist, it's difficult to understand not just the language, but the way of thinking that you find with a scientist trained a different way. And it isn't good enough just to mix them together and think that it will work. And I do not think we pay enough attention to investigating how well to communicate with people who are trained in different sorts of ways. It would seem very obvious, but what we do is we mix people together and think that they are going to be able to work together. And in my experience, it needs much more effort. So at the Crick Institute, I'm trying to start a study which I'm trying to do with the London School of Economics to see whether we can do some investigations on how to get people to overcome these particular difficulties. And one thing, sorry, I'm speaking too much maybe, one thing that we have experimented with, and it wasn't my idea, and I thought it was really stupid, was the following. Do you have speed dating in Japan? Speed dating, do you know what it is? Okay, what happens is this. Boys and girls who are interested in meeting each other sit at a table like this, and the girls and the boys go down and they speak for 2 minutes or 5 minutes with each other, and at the end they decide whether they want to have a date.
We have similar TV program.
You're too old for this, I think, Takashi. Yeah.
Sorry.
But do you have that sort of thing? Okay, well, we use speed dating between scientists from industry and scientists from the Crick, who are trained differently, and it worked. I thought it was the most stupid idea that I could imagine, but I at least tried it, and it worked. So things like this, tricks to get things to work.
ちょっと補足しとくと、クリックっていうのは
I will add some. Crick is an institute that almost the Paul made, and physicists and scientists getting together in the Cricks Institute, so where the multidisciplinary research is conducted.
For example, university we have a department, physically different place. Do you have any suggestion?
Well, again,
to promote?
At the Crick, we do not have teaching, so we have no departments because universities, I think, have to organize themselves around departments largely to do with teaching responsibilities, and that's very practical and sensible. In an institute, we don't have that need, so in fact, we have no departments, and it breaks down those barriers as well. The usual ways where people try to break down the barriers in universities are several. One is to teach combined courses, and then the faculty have to speak with each other about a combined course. That's one thing is good. Two is to put different departments in the same building and have the same coffee and places and so on, so they have to mix just voluntary, so that's a second thing. A third one is to really promote seminars and lectures that involve both types of people. And so there are things that you can do, but I don't think you can break down the departments in a university which we can in an institute.
安井先生、なんか広島大学もちょっと今
In Hiroshima University, you are working on that, right?
先生がおっしゃられたことで
As said by the Sir Nurse, what we are doing in Hiroshima University
人社系で一つの
is
大き
that
な
in the
研究家にしてはどう
we
か
are thinking
また
to combine the
自然科学
department,
あるいは
combining the
生命
very several
系ですね。そういうところで
departments in the humanity and in the
を作って
sciences.
いこうで
We
はない
may
かという
want to
ディスカッション
create
が
just
進んでい
one
る
big
と思うんですけど
department. The
まあそういう
discussion is
のを
ongoing
ですね
in that
基
direction.
盤にして
And
いけ
based
ばいい
on
の
the
か
results
な
of
と。
discussion,
た
we
だ
want
今おっしゃられ
to
た
make an
物理
effort.
的に同じビ
But
ルに入るのは難しい
it may be
かもしれ
difficult to
ない
put
です
the several departments in one building
まあ我々
physically.
もこう医学系含めて
Yes, including the medical department, we would like to work on that. As Sir Nurse said, the seminar lectures should be utilized effectively to combine the departments. And here, the panelists are well experienced in that area, but I've met some of them for the first time, so it is important to promote the combined lectures.
じゃあ次
Next, I would like to discuss several items, and we'd like to invite the views. We have the high school students and the postgraduates and the undergraduate. How we can attract the younger generation for science? Would you say a few words? The Faculty of Sciences is very involved in the science education, so I should make a comment. As Dr. Cerner said, multidisciplinary research is a direction we should head for, and I am immensely thinking of that. And, the multidisciplinary or cross-disciplinary, so it is easy to say but difficult to realize. What we want to see as the researcher here is that all the people who are going to be involved should be the experts. So we are because we do not have the study called multidisciplinary, we need experts, the mathematicians or the chemists who should be specialized in their own fields. We do not have an education to provide a multidisciplinary education because university education, as Professor Yas introduced, is now we have the reform and the graduate school, and we are working toward the direction about disciplinary education. But before that, we have to remember that each discipline should be educated well on the postgraduates. And I'd like the senior high school students to listen to me. So although we are talking about the multidisciplinary, the chemist may do the mathematics and science, or this mathematician does biology, but it is not true that you can start from the different subject. First, you have to be educated well to the one discipline you choose. That is most important. As the university education, the department faculty will provide systematic teaching for the established systematic knowledge. Then if you start research, then we should change our attitude from the conventional way of the research. The physicist may start the study in the biology, the mathematician may start life science. So we will have to promote those areas as an education, and Hiroshima University is going toward that direction. Thank you very much. Yes, this is the questions difficult to solve.
Education or multidisciplinary again. So do you have some words for this?
Well, I think science education starts in primary school, in my view, and the first thing I would say is that we don't introduce science early enough in schools. I think that children of 6 and 7 are very interested in the natural world, and it is possible to really excite them with simple science. And I think we should start very early, and it's a problem because with primary school teachers, certainly in the UK, only about 1 in 100 have any science qualifications. So
1 in 100?
Something like that. And so it's very difficult to do that. The second comment is a bit later in school, that with adolescence, often children go off science somewhat. It's difficult to do, of course. You have to learn the basic principles. And that means you need really good science teachers. It's very difficult, I think, to teach science well, and we underestimate that problem. And we need to encourage and train good teachers to keep the interest. And I think that often when there's a problem with science teaching, it can be because the teachers themselves are lacking confidence about what they are teaching. So a focus on that too. When it comes to a university, usually students have chosen to do a particular subject, so they have an interest in it. But what we shouldn't do is overwhelm them with endless facts that they should learn. And I worry in biology as the facts get more and more as we do more and more science. And I think we should have a greater focus in the life sciences anyway on principles, which I don't think we do, and then just occasional case studies that show a subject can be done in depth. So I actually think we need to overhaul undergraduate teaching as well.
Yeah. I read your biography, and you said in terms of teacher, you are in a sense quite lucky. Fortunately, you have several good teacher when you are in several steps.
It's true. I mean, at primary school, I was actually more interested in school archaeology, to be honest. But my parents encouraged me they bought me a very little telescope, just big enough to see craters on the moon and so on. So, I mean, that was very nice. So that was more from home. But I had a very good teacher that you're referring to who helped me and other pupils who were interested to do experiments outside work, outside the school day. And that was really very, very valuable, as you said.
Thank you. 今日も高校生の方も多数来ておられると思うんですけど、少なくともここに
There are some the high school students, senior high school students there, and I believe that the teachers for them must be the very good teacher to let them participate to this seminar. Do you have any further comments about the education? It's related to the education, regarding the multiple disciplinary research. Can I go back to that topic? Well, can I raise a question to Sir Paul?
Sorry, I went back to the subject of interdisciplinary research. So your Crick Institute aims to proceed the interdisciplinary or multidisciplinary researchers, including physics researchers and mathematics researchers engaged in the life sciences. We are a very small group, no comparison to your big institute, but we have been working in the similar field for 4 years in the past. But in Japan at least, there is no good opportunities to hire the researchers having good activities in the disciplinary regions. So how about the situation in England, UK, for their job opportunities for interdisciplinary researchers? It's a new rising field in science.
So I can talk about two things, both in my own experience. So the Francis Crick Institute, which is this new institute I'm director of, is a biological biomedical research institute. But as you rightly say, I wanted to involve non-life science workers in the work. But in my past experience, I found that trying to move a physicist, say, into a life science department is very difficult. Maybe because they don't think us life scientists are as smart as the physicists, because they, are the sons and daughters of Isaac Newton and Albert Einstein. So my proposal there was not to try and move them in, but to leave them, say, in the physics department, but encourage them to set up a satellite group in the Crick. So they put 2 or 3 researchers in there with specific projects. Or alternatively, we could send 2 or 3 of ours into their group to learn or to do work. So they still keep a base in their department. And that would allow us to have many more opportunities, because we're not putting in a group of 10 or 12, just 2 or 3. So I think that's a useful way of thinking how you can do it. The second thing that I got involved in was the government asked me to write a report on how research should be funded in the United Kingdom, which I did last year, or the year before actually. And I made the proposal to change the system, what we have at this moment is 7 research councils, which are all in different areas. So one of them would look after physics, chemistry, and so on. Another one, life sciences, and that's divided into medicine and agriculture and so on. And I've suggested, and they've adopted it, that although we keep the 7 research councils, we have an umbrella organization above it, which will do several things. One will be to have a stronger voice with government, because they're not dealing with 7 small bodies, they're dealing with one big body. But secondly is the point you made, that it will make it easier to fund research that falls between these different councils, and we can cut a slice of money that is absolutely committed to that type of activity. And this has been adopted by the government, and they're just setting this up now. So I'm hoping that will help in doing interdisciplinary and multidisciplinary work as well.
Thank you. Yeah, very interesting. So now,
we are the researchers and educator, but the university budget is given from the tax of the people. So how we can convert our scientific research work to the society.
The importance of the basic education of science, as well as dissemination of appropriate information to the society. 私はお酒の研究という非常にアプライドな。
I am involved in the research of brewing, and traditional way of the brewing is important, and the science is not so much relative, and that is reported by the mass media, and I feel very sorry about that. The basic science knowledge is not only for the progression of the science itself, but it is important for our daily lives. So, for instance, health supplement, which is not the evidence-based, and also the right after the nuclear accidents, and then the booing of the students would occur, and that is caused by the shortage of the knowledge, scientific knowledge in the society, and also the information dissemination is not sufficiently done. We are trying to do it, but it is not sufficient. That's what I thought. So that we are based on the tax-based budget, and so not only for the advancement of the science itself, but we have to transmit our result or outcome of our science.
Not only research, but advice to government, you said. So how can we deal with feedback to science knowledge to society?
Yes, good. I think the first thing in thinking about this is to recognize that there is a spectrum of research that all needs to be funded, and it goes from what I prefer to call discovery research. Often people call it basic research, but I prefer discovery research at just understanding how things work. And then there is activities often called translational research, where those discoveries are more mission-oriented towards an objective. And then there's applied research, which is a very particular objective. Now, all of these have common features, I mean, like doing all good science, but they also have differences in how they're organized. So particularly at discovery research, it's usually best to give a lot of freedom from the researcher to decide what is interesting and to pursue it. And you shouldn't be too top-down. On the other hand, if you're close to applied research, you have to have goals and clear objectives, and it does have to be top-down. The mistake is to think you can mix those up, that you can do discovery research top-down and you can do applied research by giving people no objectives. And often it's not recognized that you have to have a different view of all of that. And once you start to explain that, I find that some of the tensions that occur disappears, because often it usually is that governments and industry feel they have to have top-down initiatives. But once you explain that if you really want efficient discovery research, you shouldn't be doing that. They will get it. They will understand that. And I think the mistake is our own leadership, often scientific leadership, who try and take an easy course and say, oh, we are all doing translational research or applied research. And they don't bother to make the case properly in the way that I've just said. Because if we make it properly, I think people get it, politicians and public. If we just take the easy way and say, oh, you know, everything we do is useful, it isn't actually. Some things we do are useless. But by doing it, we often find things that aren't useless. So you just, because you're releasing the creativity. So it's a question, I think, of having a proper conversation. And one second small thing. When we scientists often only talk to politicians when we are complaining. And we are nearly always complaining about money. And I can tell you, if you want to put off a politician, then it's the best way to do it, to complain and only talk to them when you have something to complain about, and if you only talk about money. So I strongly think that you have to make friends with the politicians, you have to excite them with scientific discoveries, and only later do you talk about money.
いや、これは私がポール・ナース博士と一緒にいた研究所でもいつもポール博士が言ってたんです。
That's what we have been discussing at his laboratory when I was there. The scientist is only focused on to the research, but that is lazy for us. That we have to contribute to the society, and we shouldn't not always complaining the issues. So we have to change our mind.
それで今社会どうぞ。
Yes, please.
So I totally agree with your opinion that you have a friend of Japanese politician, right? And please stress the importance of basic science, okay? Not only the translational or applied science.
Thank you.
Really important. You have to make friends. Mr. Omi may understand a bit of it, but he's retired, of course.
Right, yeah. 今社会とサイエンスというかそういうの出ましたけど、まだやっぱり大事なのはやっぱり
What is most important is the human resources or the health of the human beings or the cure the disease. That's why we invite Dr. Chayama and Dr. Yasui from the medical department. The cancer as an example, that is very difficult to treat. So can I invite your comment?
I
would like to touch upon the education. As Senners said, it is important to educate science from the primary school. And I agree with you completely. So invite the children to science. It is important, but at the same time, we should promote a good understanding in cancer. And this understanding should start from the primary school to the junior high school. So how teachers teach the children about cancer? The purpose is to prevent cancer. The understanding of cancer will lead to the prevention of cancer. And in there, the children may learn the wonder of cancer at the primary school. Then he or she may want to study it. That's what we want to see. Professor Choyama, you are a researcher as well, and every day you are seeing the patients. So would you say a few comments from your clinical experience? Yes, I am working as a clinician as well. The people who are interested in science are good at collecting information, so they can be a good physician, and then that will also be contributing to their own studies. So the custom or the habit to collect information is very important.
Discovery researcher, science, but we'd like to a little bit discuss about clinic. So in our we call from bench to bed. So in the clinic, yes, that's a very big issue. But the hospital or it's real clinic side, how do you think we should?
This is crucial, of course, for the health of society, for people, and also, of course, it has potential wealth creation possibilities as well. So it's very important to get it right. We once again have the cultural problem of getting clinical doctors to work properly with scientists and scientists work with clinical doctors, and I think that requires some attention because they are trained in such different sorts of ways. What we're doing in the CRIC is we have this institute, which is large but relatively small compared with a large medical school, and we will make discoveries that sometimes will be very relevant to clinical advances. So we have links with the three main universities in London, Imperial College, University College and King's College, and their associated medical schools.
Right.
And we have established a way of working with them and with the biotechnology and pharmaceutical industry where if we think some discovery is potentially of interest for translational purposes, we will make it very easy to make that transition, because often translation is immediately made more difficult because the tech transfer company in the university and the lawyers argue and spend all the money over how much it's worth, and they spend 40% of the money when mostly it's worth nothing because it doesn't go anywhere. And so we've made a decision that we will allow it to go very cheaply into initial studies and only ask for return if something is a success.
Oh, really?
And that is a very big difference, because if you're a small startup company and you spend all this money paying lawyers, which is a big bad thing, you don't want to do that, and also it's for trivial things, like if you make this antibody, we will pay you, 500,000 yen or something. And it just puts another chain around the neck. So we get it out of the door quickly, and we just say that's a cost, and we forget about it. But if they make money on it, we want part of the money back.
Thank you. I think now time's now close to the end, but finally, very end, Paul, could you deliver your message to young people, if you can? Concluding remarks?
Yes. I've said a lot of things, but I always remember Max Perutz, who was a Nobel Laureate who lived in Britain. He was Austrian. He gave lots of advice, delivering messages, and then at the end, and he said the most important message is never follow the advice of your elders. So be careful of advice from your elders. I really only have a very simple thing to say. Doing science is really exciting and very interesting if you like doing it. And if you like doing it, then do it, and do it with a passion, and you'll get something back. Not everybody wants to do it or like doing it, that's fine too. Don't feel guilty about it. But if you like it, do it with a passion and you'll get something back. Just don't expect to become rich and famous, because that's very unlikely.
Thank you very much. ありがとうございました。
Thank you very much. Now, we'd like to conclude this session.